Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from the Department of Biochemistry. This is my second lecture about DNA replication and repair. In the first lecture, I explained you about the structure of the DNA, about the organization of the DNA, and karyotyping. Now, in this lecture, I will start with the DNA replication, the steps in the prokaryotic DNA synthesis. This is the DNA strand in which you are seeing that first of all the parental strand is separated, and from single parent molecule of the DNA, two strands or two molecules of the DNA, they are formed. Parental duplex is separated into two halves, and in each the formation of the new strand, if we see the formation of the new strand, we are noticing here that in a new strand, one is the parental strand and another is the daughter strand. This means that this process is semi-conservative replication. It means that each of the parental strands remains intact in one of the two new duplexes. In a new molecule of the DNA, one strand is the parental strand and one is the new strand. In the DNA replication, I will explain you about the replication of E. coli. Or the replication in the prokaryotes. In higher organism or in the eukaryotes, the replication is more complex, but involves the same type of mechanism. So the first step in the formation of the DNA is the strand separation. Strand separation occurs over a small region. It starts from a very small region, and that region is called the ori site or origin of replication site. This origin of replication site is the consensus sequence. Consensus sequence means the order of the nucleotide is same at each site. This is the consensus sequence, and this sequence is ori site sequence is AT rich segments, because between A and T there are only two hydrogen bonds, and the melting over the AT rich segments, they are comparatively easy. In the prokaryotes there is a single ori site, and in the eukaryotes the replication begins at multiple sites. In this diagram you are seeing that this is the DNA, and in which there are single origin of replication in the prokaryotes, and the replication bubble is formed, and after the formation of the replication bubble, the replication starts on, and it's moves on, and the two molecules of the DNA, they are formed. But in the eukaryotes, as you know that its molecules, they are large, and in order to replicate all the DNA, it needs to be originated from the multiple sites of origin. So it has many origin of replications, and after processing and completing the replication, two molecules of the DNA, they are formed. This is the again you are seeing the it's a DNA in the prokaryotes, circular DNA. This is the replication bubble. It moves on, and then finally this a single molecule of plasmid, it is replicated into two plasmids or two molecules of the DNA. This is the example of bidirectional replication. The replication in the replication fork is bidirectional. It means that when there is the formation of replication fork, two strands, one is the leading strand and one is the lagging strand, and on the both of the strand the replication starts. In the leading strand there is the continuous synthesis of the DNA, and in the lagging strand the fragments they are formed, and these fragments are called the Okazaki fragments. This is the list of proteins which are required to initiate the replication in the prokaryotes or in the E. coli. We will discuss each of the proteins one by one. All the proteins, they are not mentioned in your textbooks or in your Lippincott and Harper. There are only few proteins which are discussed. So let's move on. This is the DNA, in which you are seeing that this is the uri site, right? And at the uri site, the first protein which I will discuss and which is needed for the origin of replication is the DNA A protein. This is the DNA A protein, and this all this region which contain 3 base pair, 3 13 base pair repeats and 4 9 base pair repeats, this all region, this is the uri site. All the required proteins which are needed for the initiation of the replication, it is a group of proteins and it will form the pre-priming complex. It formed the pre-priming complex. So first of all, the DNA A protein. DNA A protein initiate the replication by binding to specific nucleotide sequence which is called the DNA A box within the uri site, right? And this, again, I have told you before that uri site will contain 80 rich sequence, and the DNA, especially the DNA A protein, it will bind at 80 rich region. Then the second protein which I will discuss here is the DNA B protein or the DNA helicases. DNA helicases is the protein, it will bind, DNA helicase or DNA B protein, it will bind to the single stranded DNA near the replication fork. Here you are seeing that this is the DNA helicase and it binds to the single stranded DNA near the replication fork and then move into the neighboring double stranded region and forcing the strands apart. It means it make the unwinding of the double helix, and it required the use of ATP. And this is the histone like proteins which are also needed in the pre-priming complex, in the formation of pre-priming complex. Okay, then another type of proteins which are needed are the single strand binding proteins. Single strand binding, these proteins, they are the single strand binding protein. The single strand binding DNA proteins, they keep the two strands of the DNA separate. The first function is that the first function of the single stranded binding DNA protein is that it keeps the strand separated, and the second function is that it protects the DNA from the nucleases. With the formation of with the separation of the strand, there comes a problem with the double strand of the DNA, is that it will cause the problem of supercoils. Yes, this diagram shows that as the DNA strand it separates, there is the formation of supercoils in the region. Positive supercoils, they are formed in the region ahead of the replication fork as a result of overwinding, right? As a result of overwinding and ahead of the replication fork, there is the formation of positive supercoils. And the negative supercoils, they are formed behind the replication fork. These supercoils, they interfere with the replication, so this problem they must be solved, and otherwise the replication cannot be proceeded, and the DNA polymerase, it will not find its way to synthesize the DNA. So to solve the problem of the supercoils, there is the enzymes which are called the topoisomerases. Okay, this diagram, it shows the telephone cord, while which shows the telephone cord, and it is tightening over the coil. Supercoiling can be demonstrated by tightening the one end of the helical telephone cord, and if it wrap around itself in space to form tightening the coil, it will be called the positive supercoils. And if the cord is twisted in the direction of loosening the coil, and the coil will again wrap around itself in opposite direction to form the negative supercoils. So this, the supercoiling of the telephone cord is similar example like that of the DNA supercoils. So the enzymes which are involved in solving the problem of supercoils are the two types of enzyme. The first type is the type 1 topoisomerase. Type 1 topoisomerase, as you are seeing in the diagram, that notice that here the one of the two strands, in one of the two strands a nick is produced by type 1 topoisomerase, and then the second strand, it will pass through this nick, and it will then the supercoils, they will be solved, or they will be then relieved. And this process, which means the production of nick and relaxation of the supercoils, it do not need the ATP, but it will use the stored energy of the phosphodiester bond, which was broken down, and then this energy will be used in resealing the strand. So transient nick is produced, and the DNA pass through that nick, and the this supercoil, it will be relieved, and finally the nick will be sealed again. In E. coli, type 1 topoisomerase relax negative supercoils, right? And in the eukaryotes and in all other prokaryotes except E. coli, it relieve both positive and negative supercoils. How does the second type of the enzyme which is used for the relieving of supercoils, it is the type 2 topoisomerase. Type 2 topoisomerase, it bind tightly to the double helix DNA. It binds tightly to the double helix DNA, and it will make a transient break in both strands, and in this process ATP is required, and it relieve both positive and negative supercoils. Also another function of the topoisomerase 2 type 2 topoisomerase is that it also it is also used in separation of interlocked molecules following replication. DNA gyrase, it is a type of type 2 topoisomerase. It introduces negative supercoils in the relaxed DNA, and which will use to neutralize the positive supercoils during the strand opening, and it will then facilitate the process of replication. So you are seeing here that the two strands, the nick is produced in the two strands, and the front half of the helix passes through the break, and which is then resealed, and I have told you before that ATP is required in this process. And DNA gyrase, it is the example of type 2 topoisomerase. There are two anti-cancer agents, camptothecins and etopsids. They target human type 1 topoisomerase and type 2 topoisomerase. They are the anti-cancer agents. They target the topoisomerases, human topoisomerases, type 1 and type 2 respectively. And there is the bacterial DNA gyrase, is a unique target of a group of antimicrobial agents called the fluoroquinolones, for example ciprofloxacin. So these are the two drugs, two anti-cancer drugs, and one I have told you, this is the ciprofloxacin. It targets the topoisomerase enzymes. Direction of DNA replication. DNA polymerase read parental strand in 3 to 5 prime direction and synthesize new strand in 5 to 3 prime direction. Here you are seeing that the replication forks, two replication forks are formed, 1 and 2, right? And this is the proceeding process. And here you are seeing that in the replication fork, and I have told you before that two types of the strand they are produced. One is the leading strand. This strand is copied in the direction of advancing fork. Okay, this strand is being copied in the direction of advancing fork and is synthesized continuously. And the second strand is the lagging strand, and it is being copied in the direction away from the advancing fork. And here you are seeing that Okazaki fragments, they are formed. In order to start the synthesis, DNA polymerase, it will not initiate the process. Rather, it will continue from the RNA primer. So the DNA polymerase, it require a short double-stranded region consisting of RNA base pair to DNA template with a free hydroxyl group on the 3' end of the RNA strand. And this RNA primer is formed by an enzyme called primase. Primase synthesize short stretches of RNA which is complementary and anti-parallel to the DNA template. Obviously, for the leading strand there will be one primer, and for the lagging strand there will be many primers. The substrate for the primer synthesis will be the 5' ribonucleoside, and after the completion of formation of the DNA, it will be removed. What is primosome? Addition of primase converts the pre-priming complex. Addition of primase converts pre-priming complex into primosome, which will make the RNA primer and synthesize leading strand and Okazaki fragments. This is how the DNA polymerase, it starts or do its work. This is the RNA primer. The blue color of the nucleotides, they are show that this is the RNA primer, and we have discussed that it uses, the DNA polymerase uses the 3' hydroxyl group of the primer and deoxyribonucleotide, DCTP. Here the DCTP is the substrate. Pyrophosphate is released. Pyrophosphate is released, and the this pyrophosphate is then further hydrolyzed into the 2-inorganic phosphate, and this DCTP is added here. below the primer. Chain elongation, I have told you before that deoxyribonucleotide, they are added to the 3' end of the growing chain. In the previous diagram I showed you that the deoxyribonucleotide, they are added to the 3' end of the growing chain. And which type of deoxyribonucleotide, which one of the 4 type is required, it will be dictated by the template strand. Okay, now the DNA polymerase specifically used for the DNA synthesis is the DNA polymerase 3. DNA polymerase 3, it uses 3' hydroxyl group of the RNA primer. It is highly processive enzyme. This enzyme is highly processive enzyme. Processive enzyme means that this enzyme, DNA polymerase 3, it remains bound to the template strand as it move along and does not diffuse away and then rebind before adding each new nucleotide. So it will remain bound until the whole of the DNA molecule it is synthesized. This processivity is due to the beta subunit of the enzyme, which serve as a sliding DNA clamp, right? And this clamp then move along the DNA strand and going on starts and then continues on synthesizing the DNA strands. This diagram, it shows actually all the steps of the DNA synthesis. This is the DNA helicase. This is the DNA helicase, the green color. It is a DNA helicase and it is moving in advancement of the replication fork. And this is the leading strand. This is the leading strand, and this blue color fragment, this is the RNA primer, and this green color enzyme, it starts from the RNA primer and it is synthesizing in a continuous manner on the leading strand. And these two proteins here are showing are the single stranded DNA binding proteins. And here in the leading strand you are seeing that there is only a single RNA primer. Now come to the lagging strand. In the lagging strand, the RNA, this is the primase, this bluish color protein, this is the primer, and you are seeing here that 2 primers, they are synthesized, and the fragments, the new fragments or the new complementary fragments, they are synthesizing away from the advancement of the replication fork. So there are many fragments, they are formed, and with the advancement of the opening of the strands, there will be the formation of supercoils, which are removed or which are relieved by the topoisomerase enzymes. This diagram shows the activity of the 2 types of activity of the DNA polymerase. 2 types of activity. One is the which we have already discussed, 5 to 3' polymerase activity. Let me tell you that the strand which is the parental strand which is readed by the DNA polymerase is in the direction of 3 to 5' direction, and the complementary new strand which is formed is in the direction of 5 to 3' direction. So the first type of activity of the DNA polymerase 3 is 5 to 3' polymerase activity. Here an incoming nucleoside triphosphate is correctly matched to its complementary base on the DNA template. On the DNA template there is the C, and the incoming nucleotide is the GTP. and this G, it will be paired and it will formed with the C, forming the 3 hydrogen bonds, and this activity is the 5' to 3' polymerase activity. Now come to the second type of activity. This is the 3' to 5' exonuclease activity. This exonuclease activity, it means that if the bases, they are mistakenly not correctly base paired. For example, here the A comes in front of C, which is wrong. There must be G. So this A, it will be removed by the 3' to 5' exonuclease activity. Right? This exonuclease activity, it will excise the mismatch nucleotide, and again then the 5' to 3' polymerase activity will bind the correctly matched nucleotide. Finally, after completion of the complementary strand, there comes the removal. There comes a process of removal of the primer. Finally, the primer, RNA primer, it must be removed, and this removing is done by the DNA polymerase 1. DNA polymerase 1 is the enzyme. It is a slightly different from the DNA polymerase 3 because it contains 3 types of activities. The 2 types are same, which is the 5' to 3' exonuclease activity and 3 to 5 exonuclease activity. It contains an additional activity, which is the 5' to 3 exonuclease activity. This activity is not present in DNA polymerase 3. So DNA polymerase 1, it can replace a segment of RNA or DNA paired to the template strand in the process known as Nick translation. This is how the RNA primers in the Okazaki fragments, there are 3 RNA primers. They are present in the Okazaki fragments. They are excised by the DNA polymerase 1, and then this gap, the gap which is produced by removal of the RNA primer, it is again filled by the DNA polymerase 1. Finally, after this gap is filled, the final 3 to 5' phosphodiester bond, it is sealed by the DNA ligase. This shows the final phosphodiester bond, which is sealed by the DNA ligase. And the process is, again, it requires the ATP, and here again the pyrophosphate is removed. I have told in my lectures many times that production of pyrophosphate PPI with subsequent removal, with subsequent hydrolysis of the PPI to 2-PI is a common theme in the biochemistry. Removal of or production of pyrophosphate product, it derives the reaction in forward direction, making it essentially irreversible. So the reactions in which the PPI or pyrophosphate is removed, they are irreversible reactions. Finally, there comes the termination process. This termination process is due to a sequence specific binding protein TUS, terminus utilization substance, to the replication termination site TER site. So TER site, it binds to the TER site, and this binding, it makes or it stops the working or the movement of the DNA polymerase. So this was all about my today's lecture. In the next lecture, I will explain you about the eukaryotic DNA replication. So best of wishes, good luck, Allah Hafiz.